Okay, well, firstly, I'd like to start by thanking the organisers, particularly Professor Szilard, who invited me and always puts on a great meeting. I've changed my title slightly to concentrate more on the sort of aspects of our work which looks at cognition, particularly memory and learning, and in particular in our animal models and also some recent human intervention trials which we've done with cognitive endpoints. So the first slide really illustrates the problem, and we've seen this. I mean, the slide on the right, on the left-hand side, seems like a very nice story. We're now keeping people alive for much longer. We're all going to live longer, which is great. However, if you look at the right-hand side, you'll see that as we do get older, we fall into categories of risk where we're more likely to develop dementia, particularly of the Alzheimer's type, vascular dementia, and even dementias associated with Parkinson's disease. So you see risk factors for these two diseases markedly increase as we age. So there's been a lot of interest in natural compounds, many of which have been discussed here already. I'm going to concentrate on our work with flavonoids today, but there's been some very nice work coming out of David Smith's group in Oxford looking at 3B vitamins and their effect over a 2-year period on brain atrophy and also related cognitive function deficits, which have been very interesting. Quite a lot of work with curcumin, which is found in turmeric, very interesting sort of dimer of ferulic acid, another phenolic acid, which is similar in some ways to some of the phenolic colonic metabolites of flavonoids. And also, of course, there's been a long interest in omega-3 and brain function, particularly during development in the womb. So with regards to flavonoids, I've worked for some years in this area, and there's been a lot of discussion over the years about how they may function. So initially, there was a lot of work being done on the potential antioxidant effects of flavonoids. However, after a series of experiments which investigated their absorption and metabolism, showing that they're heavily metabolized, they're not greatly absorbed in vivo, there was more of a focus on more physiological effects. So, for instance, there's been a lot of work recently from the cardiovascular field looking at their effects acutely and chronically on blood pressure, on markers of vascular function, particularly FMD and LDI. And also there's been, of course, a lot of interest from other fields like cancer, inflammatory diseases, and also neurodegeneration in general. So the work I'm going to talk to you today is about aging, and it's about aging of the brain and what happens when the brain ages, and how diet can modify some of those effects which occur with aging. So there are quite well described cellular and molecular changes which occur, particularly in regions which control memory and learning within the brain. The regions of interest, for example, in humans are the hippocampus, perhaps more related to short-term memory, and the neocortex, which is more involved in long-term storage and processing of memories. And so these molecular changes are very well correlated with behavioural aspects. And so with all the work that we do at Reading, we try to sort of understand all of these processes and how they fit together, the different levels. And this, of course, involves more expertise than just a nutritionist or a neuroscientist or a psychologist. I think it really involves all of these people coming together. And it also requires an understanding of how these processes work at the cellular and molecular level. So we have a number of different model systems set up at Reading, which range from single-cell experiments, really looking at neuronal cultures, co-cultures with glial, astrocytes, microglia and neurons, either cortical, striatal neurons, and trying to understand how they work mechanistically. So we try to use dietary levels of these compounds and their metabolites, and we look at how they change signaling pathways which are involved with memory and learning. However, today I'm going to concentrate on some of the studies we've been running in the animal models and also the human studies, like I mentioned. So in terms of the rodent models that we use, we mainly use the 8-arm radial maze with young animals, 1 to 6 months. And in older animals, we use the 8-arm radial maze, but we run a T-maze alternation task because obviously aged animals are not so cognitively active, and so they're unable to perform very well within the more complicated 8-arm maze environment. And we have two paradigms. One where we feed during consolidation and retention, which we think more closely looks at whether the diet has an impact on memory processes itself, so the recall of stored information. And we also have a feeding paradigm, which is the lower one, which looks at what happens during task learning, which is more of a sort of learning and the laying down of the task. So the first series of experiments we did was over a 12-week feeding period where we fed an 18-month aged old rat with a 2% blueberry diet. And you can see that, is there a pointer on here? Yes. This is the performance of the old animals compared to the young animals. And this is the aged group over a 12-week period after fed with blueberries, 2%, incorporated in the diet, whole, ground up, freeze-dried, and put in. So this is a kind of, in some ways, a repeat of Jim Joseph's work, although slightly different feeding paradigm in that we use the whole blueberry with all the skins rather than just the juice. But you can see after a 3-week period, there's almost a complete recovery of the cognitive performance of the old animals within this testing paradigm. And this also correlated with the time to make a correct choice within the maze environment. So we were particularly interested in trying to understand how those effects were mediated, those behavioural effects were mediated. And of course there was a lot of work that had been done over the past 50 years, really trying to understand how memory is laid down within the brain. And it's known that during acquisition of memory, there's a short-term post-translational modification of proteins, phosphorylation of kinases, particularly MAP kinases signalling to Krebs. And also for storage and long-term memory, it requires new protein synthesis. And we'd also previously done a lot of work where, this was in the cellular work, where we were able to show that actually flavonoids and their metabolites at physiological concentrations are capable of increasing the phosphorylation of an AKT towards Krebs. And this signals on to certain neurotrophins and causes changes in synaptic plasticity within the cellular model. Also, you can see that they inhibit brain aging potentially by downregulating or inhibiting the phosphorylation of the pro-apoptotic J and K. So we were interested to know what was going on in the molecular sense in those animals where we saw such fantastic behavioural responses. And without going into all of the data, we found modifications within the hippocampus, but not within cortical regions, of Krebs phosphorylation, ERK phosphorylation, leading to an increased expression of BDNF, which is a neurotrophin which has long been associated with the sort of the learning and synaptic plasticity long-term potentiation paradigms. We also saw changes in proteins which were involved with enhanced protein synthesis within synapses, increasing the number of dendritic spines and also the size of dendritic spines, so making communication between neuronal partners much more efficient. And there was very good correlations with Krebs phosphorylation and BDNF levels within the hippocampus with the behavioural performance. But you start to see increases, particularly for anthocyanins, within the dendrite gyrus and the CA1 region. And in the case of the in the CA3 region, in the case of the CA1 region, this blueberry effect, which was the effect I showed on the last slide in terms of the raw data, is close to significance as well. And lastly, from the animal point of view, we also carry out some Morris water maze models where we also saw improvements with 2 doses, 2% and 4%. And actually, these were quite nicely correlated with changes in polysialylated NCAM, which has been long associated with synaptogenesis, new synapse formations within neuronal clusters, and also with neuronal repair. And so, I mean, to summarise that, we're quite advanced now in understanding how particularly flavonoid and flavonoid metabolites might mediate changes in neuronal morphology. And we do think that some of this might be related to vascular effects, and that becomes particularly apparent when I show the human data in a few seconds. With particular regard to these vascular effects, there's long been known from Professor Gage's lab at the Salk Institute that vascular changes within the hippocampus are associated with neurogenesis. So there's a population of stem cells within the hippocampus which can respond to vascular factors and, for example, nitric oxide. So when we come to testing in humans, of course it's a lot more difficult, it's a lot more complicated, there's a lot more higher functions you have to consider. And so it's a lot more, you can't just stick people in maze environments and try to assess their spatial memory. You also need to consider things like attention, executive function, episodic memory, motor skills, even things like mood. And of course some of these can be assessed by simple visual analog scales, but others require a battery of cognitive test conditions which may be primed to induce fatigue over the day of testing, and so you can look for recovery from fatigue on some of those, or induce stress and look for a recovery from stress due to diet. But they're also sensitive to look at changes in the functioning of individual tasks, but also by obtaining Z-scores from each individual task, you can bring them together and have an overall score for particular domains of cognitive function, or cognitive function overall. And you can see that here. So things like the MMSE score, which are used clinically for the assessment of Alzheimer's disease, are not really any good for us here because they're just not sensitive enough to changes in diet. So there's questions sort of like what day is it, what year is it, what month is it. Quite serious deficits in cognitive function need to occur before you start to see changes there. But we can use it for screening to make sure there's no one who's seriously cognitively impaired. So we use a different series of tests which may be, I mean, I'm not a psychologist, so I won't go into specific details of these tasks, but they do map on to certain constructs or domains of cognitive function, which are correlated to activity in specific regions. So some of them will be particularly active in frontal regions or hippocampal regions. And we can assess things like executive function, verbal recall, recognition, spatial working memory. And some of these are very sensitive to aging and are some of the first ones to you'd see deficits in patients who are mildly cognitively impaired. The first series of experiments we did was actually with a blueberry, so we continued that on. And we used a sort of similar dose, 160 grams, with a control diet which was isocaloric and matched for things like vitamin C, minerals, and certain carbohydrates. And this was an acute study, so we just looked over the day. And the reasons for doing that is because we already had some quite nice vascular data with FMD as the endpoint, which suggested over a 1 to 5 hour period there's quite a nice change in vascular activity in response to blueberry flavonoids. And so we did this in 2 subsets with young and old, and if anything we saw actually more of a response in young individuals, and this may reflect the health of the vascular system in these individuals, if indeed it is being mediated by the vascular system and blood flow to the brain. So in the control diet over the 5 hour period you see a slight reduction in attention. This is a test which measures attentional performance over the day, and this is completely expected. We didn't see actually an increase in response to the blueberry diet, but it was just like a maintenance of function over the day. And this was also apparent in the older individuals, but if anything it was less apparent, and of course we did see differences between young and old at baseline. So that was perhaps not as well controlled an experiment as we would have liked. But we recently done another experiment with 63 healthy older individuals with a much more controlled diet. So we had a high cocoa flavanol diet, around about 500 milligrams, and a controlled low flavanol diet, only 29. And particularly important, it was matched for things like caffeine and theobromine, which are of course present in cocoa. We did both an acute and a chronic study, and I'll show you the data from both. So I mentioned that you're able to clump together these tests and obtain Z-scores, and this allows you to compare functioning between or people's performance on each task. And so if you take an overall Z-score from 5 executive function tasks, you can see that there was a significant improvement within a 2-hour period after the high flavanol cocoa intervention compared to the low flavanol control. And with episodic memory, which was a Z-score of 4 functioning tasks, you see a deficit. This is completely normal over the 2-hour period in episodic memory. And this is attenuated by the high flavanol diet. So in some ways, it's very similar to what we saw with the blueberry intervention. Is to try to assess what's going on in terms of brain activity. So we recently acquired an fMRI, a 3T fMRI scanner, and we're able to do now arterial spin labeling, which looks at changes in blood flow, cerebral blood flow to the brain in response to diets or drugs. And also functional MRI, where you can look at hemodynamic changes in response to specific test activities. So people would actually perform those cognitive tests within the scanner, and you're able to see what's happening to blood flow during test performance. So it's kind of like a physiological marker for the behavioural activity. And that was interesting to us because initially we didn't find any chronic improvements in we didn't see any significant changes within the same group of individuals as we saw the acute changes but chronically over a 12-week period. So we're quite disappointed, but on the basis of the previous data, we decided to split our cohort into different tertiles of intake. And we were able to see that then actually overall executive function task scores were close to significantly improved within the lowest tertile intake, and some individual performance on tasks. This is a letter memory performance task where you have to remember a series of letters and words. You can see only significant improvement was seen in the lower intake group, where individuals who came onto the study who had fairly low intake of flavanols and procyanidins, they got the most benefit from the intervention. So I think this is my last slide. I just wanted to highlight where we think we are with this at the moment. So we're really working at 2 different levels. So we're still not completely clear what's actually mediating the effects of flavonoids in vivo, or flavanols in vivo. Of course, it's most likely to be a metabolite or a metabolic form, either a glucuronide, a sulfate, a methylated glucuronide, or perhaps one of the colonically derived metabolites, which have to date really not been investigated that well. But we think that we can measure them in the brain. It's quite difficult to do that. You need quite sensitive technologies to do that, but we have been able to measure some levels of flavonoid metabolites increasing in the brain after feeding. And they seem to have effects on existing neuronal infrastructure, if you like, changing synaptic activity, leading to synaptic plasticity. In the experiments I showed you, we're also now looking at changes in the numbers of neuronal spine connections. You can also look by a series of electrical arrays at actual communication between neuronal partners and neuronal clusters. So that's one side of the equation, but we also think either through peripheral vascular effects or direct cerebral vascular effects, they can also have an effect on increasing the blood flow to the brain. And this is much more likely to be underpinning the acute activity of flavonoids on things like attention and episodic memory. Whereas this side of the equation is much more likely to be inducing more long-term effects. Having said that, of course, as I mentioned, any changes in blood flow to regions of the brain, but particularly the hippocampus, have been associated with angiogenesis and also neurogenesis. And neurogenesis is particularly interesting because, of course, if you can induce new growth of neurons, then you can potentially repair damage. And there's also a group of people who actually think that neurogenesis actually is very important, not only for repair of damage, but for cognition generally. And so it's something which is something we're interested in looking at in future. And by using the MRI, we can actually look into the brain and look for specific markers of neurogenesis in humans. So I'd just like to finish off by thanking my group, but in particular Caterina Rendero, who's here and has a poster, who performed the majority of the animal work that I showed today. And so I really encourage you to go and speak to her later. I think her poster is number 2, so almost outside the whole building. And also, where is she? Caroline Saunders, who conducted the human study. And thank you very much.
You focus on anthocyanin from blueberries and on flavonoids.
Mm.
Does any kind of flavonoid family that doesn't work on this? Because this would be helpful to understand the structure-function relationship.
Yeah.
And the second question is regarding the vascular, the blood flow.
Yes.
Is there any nitric oxide involvement in the effect you've observed?
Okay. Well, I'll take the last part first. I mean, in terms of the peripheral vascular effects, people have measured nitric oxide at the same time. They've measured metabolites and they've measured F&B responses, and certainly nitric oxide's increased. Whether or not that's through direct stimulation of eNOS to produce more nitric oxide, or through perhaps an inhibition of NADPH oxidase, which reduces superoxide, which knocks out NO, I don't think people really know. The only thing I would say about that is if the most potent NADPH oxidase inhibitors from flavonoids are actually colonic metabolites, and you don't get them until about 4 hours. And so in terms of the vascular responses at 2 hours, I would think it was more likely to be due to their effect on the activity of eNOS and the production of NO. With regards to the actual activity of different groups, it's a difficult one. I mean, what I didn't say was that the activities of the flavonols and the anthocyanins that I've shown were not dosed at the same levels, actually. One was at 2 milligram and one was at 5 milligram, and that was to reflect what you find in blueberry. But we didn't see really any significant differences in the activity between the two. Now, it would be nice to continue and look at equal doses of an example compound from each flavonoid subgroup, so to sort of look at whether or not one group has a better activity than the other. But we've not done that yet. We've done some work with flavonones on the peripheral vascular responses, and they certainly have a lesser effect than flavonols. So we perhaps would expect less of a cerebral change in blood flow and a differential response on attention as well.
I enjoyed your talk. I have this question. Is it possible to look at flavonoid metabolic compounds in the urine of the humans or the animals you treated with NMR in order to identify the metabolites?
Well, it's certainly possible to measure metabolites of flavonoids in urine and also in blood, actually. So where am I looking? Did you mean specifically during the MRI experiments, or to link it up with activity within the brain, or?
Assuming that the effect is given is due to metabolites.
Yes,
it would
yes,
be extremely
yes,
interesting to identify
yes,
them. So
yes.
if you have people treated or animals treated and non-treated, and
Yes.
you look at urine or plasma,
Yes, yes.
it could be interesting to see if there are some peaks or some signals in the spectra.
No, and
with NMR in order to identify them.
we have done that with the peripheral vascular response. So there's a correlation with activity for FMD with epicatechin itself, and also one of the metabolites, the 3'-methyl-7-glucuronide. We're also planning to do that with the cognitive activity, but we're just developing the method at the moment so we can assess a greater array of metabolites. At present, we're only looking at 4, so it'd be nice to have all of them, so if we, in case we miss a sort of like a mediating compound.
Very short.
Yes.
The gentleman first, the lady.
Thank you, very nice presentation. My question is, I have two short questions. The first one is, when you look to the cognitive function and BDNF correlation in animal and in humans, did you also collect any data on physical activity level after the intervention? And the second one is, with respect to the last study in humans, you correlated the BDNF increase in plasma actually and cognitive function with BDNF expression in the brain. But can you exclude that this increase is due to the expression from different kind of cell type? Thank you.
No. Well, with regards to the latter point, no, definitely not. I mean, we're not knowing where that is coming from, being expressed from. With regards to the first point, I mean, there's been quite a big literature on exercise and BDNF and increased cognitive function. And so we were very careful, obviously with the rats it's very controlled. We did also do some motor performance tests, and that shows significant increases. But during the feeding period and the testing period, there was no differences in their physical activity between the groups. In the human study, which was a randomised crossover controlled study, we also screened for people who undertook, I think it was around about 2 hours of exercise per week. So there was no overall differences in the levels of activity. Of course, with human trials, you're always, particularly chronic trials, you're always limited in what you can get from them. But certainly we tried at best to control for exercise because we knew it may also have an effect on cognition.
Very short.
Thank you for this next presentation. Yes, the difference with the low flavanol and high flavanol on blood pressure, is it related to the biodisponibility of the drug or other mechanisms? Because we have made some study flavonoids from pomegranates in spontaneous hypertensive breeds, and when we give by oral administration or by intravenous, we have difference in blood pressure.
So you were saying, did the low have a different effect on blood pressure to the high? Is that?
Yes, the mechanism.
The
disponibility of these products or other mechanisms.
On other mechanisms of generation of nitric oxide.
Yes.
Well, we haven't really looked into that in too much depth within the human trials we've done, apart from measuring nitric oxide by chemiluminescence technologies. There is a big interest in understanding how exactly flavonoids might be able to, or flavonoid metabolites might be able to increase nitric oxide. The problem is when you go into using cellular systems for that, even primary endothelial cells are sometimes quite difficult to work with and quite difficult to get responses, and we're not really experts in that area, so we haven't sort of pursued that. But I think it's very interesting. And certainly other foods which contain those compounds are just as likely to have an effect.
Yeah.